Hi, in this video I'm going to be talking about transduction in the gustatory system. We can detect 5 different tastants: salt, sour, sweet, bitter, and umami. But salt and sour have a differing transduction mechanism than the other 3. In our other senses, the transduction mechanism is the same within each sensory system. So why might taste have multiple transduction mechanisms to send the same type of information to an electrical signal? First, let's review some anatomy from class. On the surface of our tongue, we have raised bumps called papillae. These papillae create small grooves which compounds from our food can get stuck in as we eat. On the sides of these grooves, we have our taste buds, which can detect these compounds. Each taste bud is a clump of different receptor cells. Under the label line hypothesis, each receptor only accepts one unique tastant. But under the population coding hypothesis, a single receptor will respond to multiple tastants at differing levels. Now that we're oriented to our location in the body, let's zoom in to a single salt receptor. So just one of these long pink receptor cells. Chemically, a salt is an ionic compound that dissociates into positive cations and negative anions. Our cells already have specific ion channels that can transport sodium into the cell. So when we eat something salty, sodium ions can move right through these selective channels. As sodium increases, a graded potential of corresponding serotonin is released into the synaptic gap between the receptor cell and a gustatory afferent neuron, resulting in an electrical signal. Sour foods have a high acid content. Chemically, this just means that a molecule can easily donate a proton or a hydrogen ion. In this example of citric acid, you can see the carboxylic acid group outlined in red. And as you might remember from chemistry, a carboxylic acid group readily donates the hydrogen atom, or the proton, because of the high affinity for electrons of the oxygen. So this hydrogen atom can subsequently find a sour receptor in a taste bud. Just like in our salty receptor, hydrogen ions can directly interact with a proton-selective ion channel to flow into the cell. But rather than directly leading to depolarization of the cell, they stick to the end of the potassium-selective channel, which blocks the channel. When potassium can't leave, a buildup of positive ions in the cell leads to a similar depolarizing graded potential, resulting in serotonin release in a synaptic gap between the receptor cell and a gustatory afferent neuron. Now, bitter, sweet, and umami tastants do not immediately interact with the ion channels, and this makes sense. These tastes are often produced by large chemical compounds, often with aromatic rings, things like caffeine, sucrose, and glutamate. They're bulky and can't easily pass through the membrane or through a selective channel. So instead, these tastants utilize a pathway similar to olfaction with G-protein coupled receptors. In this pathway, tastants bind to our receptor proteins which sit on the extracellular membrane of the receptor cell, allowing the outside of the cell to communicate with the inside of the cell. When the tastant binds, the receptor protein undergoes a conformational change, which activates the G protein inside the cell to then bind and activate an enzyme. This enzyme then catalyzes a series of reactions to open up sodium ion channels, which allow sodium outside the cell to flood in, resulting in another depolarizing graded potential. ATP then flows out of the cell into the synaptic gap to communicate with gustatory afferent neurons. So this is how these chemicals are converted into an electrical signal. But how are the afferent neurons arranged so that we register different tastes? As it turns out, this is quite difficult to study. In the label line hypothesis, a receptor only accepts one tastant, which synapses on a gustatory neuron. That signal transmits through the brainstem, then through the thalamus, and finally to the corresponding region of the gustatory cortex. But under the population coding hypothesis, there's a lot more interaction. Receptors respond to combinations of tastants, which then synapse on multiple gustatory afferent neurons. Adults have between 2,000 and 4,000 taste buds, with even more receptors. So this hypothesis suggests that our interpretation of taste comes from unique patterns as the signal moves to the brain through higher level processing.